Hi all, we are now looking at question number 24 from GATE Biotechnology 2022 paper. So the question is, which of the following is not used for generating an optimal alignment of two nucleotide sequences? Option A, gap penalties. Option B, match scores. Option C, mismatch scores. And option D, nucleotide composition. So, before we get into this, let's look at some information about sequences, their alignment, why it's important, and how it's done. So, we all know that the human genome is 3.2 billion bases long and it's composed of 4 nucleotides: adenine, thymine, guanine, and cytosine. So, this is equivalent to the letters in more than 2000 books taken together. Bioinformatics is the study of these letters of the genome using computational methods. It is used to study the genome at the DNA level, the transcriptome at the RNA level, and the protein sequences to understand molecular basis of diseases, to understand how these sequences form secondary structures to function, and to understand the trends or patterns in biological data, like how these letters are different in people with higher risk for a specific disease, and in phylogenetics to study evolutionary relationships. So, how do we get to know those sequence structures? So, we have several sequencing technologies now to read the nucleotides and provide the sequence readouts in computer readable formats that can be converted to human readable formats too. The sequencing technologies have evolved from first generation Sanger sequencing, then the next generation short read Illumina sequencing, and the third generation long read sequencing like Oxford Nanopore technology that is faster and it's used to read longer sequences. We would get into the details of these technologies in some other session. So, several large scale sequencing projects have sequenced thousands of people's genomes across the world, which helped assemble the human genome. And now we have information of more than 90% of our genomes, although the functions of majority of the genome is not known, given the fact that only less than 3% code for proteins. So yeah, all these to uncover these sequence information so that we could make biological sense out of it. So some of the large-scale genome sequencing projects are the Thousand Genomes Project, the Earth Biogenome Project, the Vertebrate Genome Projects, and so on. So for all this, sequence alignment is a primary and most important task in bioinformatics. For example, in sequence assembly, where we get our genome sequence, these would be just meaningless fragments of nucleotide reads. They make sense only after we assemble them or align them with the reference genome so that we know which read maps to which parts of the annotated regions of the genome. We would get to know any aberrations or mutations on the genome this way, and we could understand the genetic basis of diseases. This is just one example where sequence alignment is important. It is further used for functional prediction, evolutionary analysis, and so on. So let us now understand some terminologies often used in sequence alignments or pattern matching. So one of them is homology. So genes that are derived from a common ancestor gene are called homologs. Orthologous genes are homologous genes in different organisms. And paralogous genes are homologous genes in one organism that derive from gene duplication. So, gene duplication is when one gene is duplicated in multiple copies that therefore free to evolve and assume new functions. So, here's an example. The bones in the forelimbs are color coded to indicate similarities in structure. They are all evolved from an ancestral tetrapod. We can see that the crocodile's forelimb looks more structurally similar because it is closest to the ancestor. So, structures like these that have a common origin but may not have functional similarities are called homologs. So, here are just more examples of the structures of limbs across various species. And moreover, analogous genes are genes that have identical or similar function but don't share a common ancestor and are therefore unrelated as opposed to homologous genes as we saw earlier. For example, the genes encoding for the tail and the flap of loose skin between the limbs that enable flight in flying squirrel and flying phalanger are different species, but they have the same functionalities. And another example is the snout of different species of anteaters, and so on. Further, the phenotypical or structural similarities arise from a very small percentage of dissimilarities in the genome. For example, chimpanzees are genetically closest to humans, and in fact, chimpanzees share about 98.6% of our DNA. Only 1.4% change in the genome is responsible for the whole range of dissimilarities. So, how do these dissimilarities arise? So, such dissimilarities arise by mutations. So, mutations are where a nucleotide at a certain location is replaced by another nucleotide. For example, like nucleotide G in place of T in the original sequence. So, this is a simple example of mutation at a single nucleotide level called single nucleotide polymorphism, abbreviated as SNP. It's pronounced as SNP. Further, we have insertions where at a certain location, one new nucleotide is inserted between two existing nucleotides. For example, like G that is not supposed to be in the sequence between the two A's in the original sequence has arisen over here. Then we have deletion. Deletion is where at a certain location an existing nucleotide is deleted. For example, the T in the original sequence is missing. So these insertions and deletions are often commonly termed as indels. Further, similarity between two or more DNA sequences is achieved by finding a good alignment between them. Sequence alignment is an arrangement of two or more sequences highlighting the similarity. The sequences are padded with gaps represented by dashes so that wherever possible, columns contain identical characters from the sequences involved. Sequence alignment is used to study the evolution of the sequences from a common ancestor, such as protein sequences or DNA sequences. So, mismatches in the alignment correspond to mutations, and gaps correspond to insertions or deletions. Sequence alignment also refers to the process of constructing significant alignments in a database of potentially unrelated sequences. Now, pairwise sequence alignment methods are connected with finding the best matching piecewise local or global alignments of protein, that is amino acids, or DNA, which are nucleic acid sequences. Typically, the purpose of this is to find homologs, that are relatives of a gene or gene product in a database of known examples. This information is used for answering a variety of biological questions, like the identification of sequences of unknown structure or function, the study of molecular evolution. Now, looking into global alignment. A global alignment between two sequences is an alignment in which all the characters in both sequences participate in the alignment. Global alignments are useful mostly for finding closely related sequences. As these sequences are also easily identified by local alignment methods, global alignment is now somewhat deprecated as a technique. Further, there are several complications to molecular evolution, such as domain shuffling, which prevent these methods from being useful. Needleman-Wunsch algorithm is commonly used for global alignment. Here's an example of global alignment between two protein sequences. Global alignment tries to find the best fit between two sequences. A higher score would be achieved if sequences are aligned this way. The Needleman-Wunsch algorithm is the commonly used algorithm for global alignment. It performs a global alignment on two sequences S and T and is applied to align protein or nucleotide sequences. The Needleman-Wunsch algorithm is an example of dynamic programming and is guaranteed to find the alignment with the maximum score. So the cost of aligning two symbols x and y is the scoring function given by this equation. The cost of the entire alignment is the sum of scores of alignment at each position. Here is a simple scoring function where insertion or deletion and mismatches are given a score of -1 and matches are given a score of 1. However, the scoring values may differ with each algorithm used. With that logic here, let's consider the same two protein sequences for alignment. Here in this sequence, we have 7 matches, 2 mismatches, and 3 gaps. So based on the scoring logic, we have 7 multiplied by 1 and 2 multiplied by -1 for the mismatches and 3 multiplied by -1 for the gaps. So the total score for this particular alignment is 2. So the optimal global alignment between two sequences is the alignment that maximizes the total alignment score over all possible alignments. Finding the optimal alignment looks like a combinatorial optimization problem. It generates all the possible alignments and then it computes the score matrix and then selects the alignment with the maximum score. Here is an example for two nucleotide sequences to be aligned. More than one alignment may be possible, so how to choose the best of them? So like we just saw in the earlier slides, we compute the scores for each possible alignment and decide the best alignment. A different scoring system is used here where match score is 1, mismatch score is 0, gap score is -2, and if there is a gap extension, that is continuous gaps, the score is -1. So let's consider the two nucleotide sequences, and there are two different possible alignments. So in the first possibility, as seen here, we have 17 matches, 2 mismatches, 3 gaps, 1, 2, and 3 gaps, and 4 gap extension, 1, 2, 3, and 4 gap extension. So multiplying each of these with the relevant scores, we get the total alignment score to be 7. So now let's consider the next possibility. In this case, we have 17 matches, 2 mismatches, 2 gaps, and 5 gap extensions. So calculating the score, we get an overall score of 8. So the second possibility is considered to be the best alignment of these two nucleotide sequences. In general, the scores of the multiple alignment probabilities are computed in the form of a scoring matrix. Examples of these scoring matrices are PAM and BLOSSOM matrices. Let us now look at an example of a scoring matrix. We have nucleotides of two sequences as the rows and columns of the matrix respectively. We add dots against the matching bases. Like against A in the first column, we put a dot against all A's in each row, and in a similar manner for every other base in the matrix. We could see a pattern here. So following this pattern would be an optimal alignment. It is read by following the pattern as 4 gaps here in the first sequence, followed by C, T, G, A, T, A, and we can notice that there's a mismatch here at this particular base. So it is read as A here, while in the other sequence it's T as shown here. And then we again follow the rest of the nucleotides, and here we could see that there are deletions. There are 3 deletions in the column, which means there's a deletion in the second sequence. So in this way we get an optimal alignment. In alignment algorithms, instead of those dots, we would have scores, and the alignment is read by following the path of the highest score along each position. We just learnt about global alignment. Now let's just have an overview about local alignment. Local alignment methods find related regions within sequences. That is, they consist of a subset of characters within each sequence. For example, positions 20 to 40 of sequence A might be aligned with positions 50 to 70 of sequence B. This is a more flexible technique than global alignment and has the advantage that related regions which appear in a different order in the two proteins, called domain shuffling, can be identified as being related. This is not possible with global alignment methods. The optimal local alignment between two sequences, S and T, is the optimal global alignment of the subsequences for some optimal choice of I1, I2, J1, and J2, which are the boundaries of the subsequences. So here's an example of a local alignment where a subsequence aligns to a larger sequence. And for a better understanding, we could see that global alignment is the alignment of two complete sequences, while local alignment is the alignment of a subsequence to a subset of a larger sequence. The Smith-Waterman algorithm is for determining similar regions between two nucleotide or protein sequences. Smith-Waterman is also a dynamic programming algorithm and improves on Needleman-Wunsch. As such, it has the desirable property that it is guaranteed to find the optimal local alignment with respect to the scoring system being used. However, the Smith-Waterman algorithm is demanding of time and memory resources. As a result, it has largely been replaced in practical use by the BLAST algorithm. Although not guaranteed to find optimal alignments, BLAST is much more efficient. So multiple sequence alignment is where you align two or more sequences. We compare all the sequences pairwise, followed by cluster analysis to generate hierarchy of the pairwise data. So BLAST, which is the abbreviation for Basic Local Alignment Search Tool. So it's a tool used to compare gene and protein sequences against other sequences in public databases. So there are several types of BLAST. So BLAST basically helps us do multiple sequence alignment where a single query, a sequence of interest, is matched and aligned against multiple sequences in public databases. For example, BLAST-N is used to query a nucleotide sequence against a database of other nucleotide sequences. And BLAST-P does a protein-to-protein search, BLAST-X for nucleotide against proteins, T-BLAST-X is for translated nucleotides against translated nucleotides, and T-BLAST-N is protein sequences against nucleotides in 6 open reading frames, and MEGA-BLAST is for large number of query sequences. So BLAST, why do we need to use BLAST? So BLAST is basically used to identify species, to locate domains, to establish phylogeny, and for DNA mapping and for comparison. So BLAST is available as a web-based interface and also in the form of a command line interface. So there are certain parameters that BLAST tells us which we look at to choose the optimal alignment, and one of them is the E value, which is the expectation value. The expectation value is nothing but the number of hits expected by chance when searching a database of a particular size. So smaller the value of E, the more significant is the match. So the value of E decreases exponentially as the score of the match increases. And there's another parameter called bit score. It measures sequence similarity independent of query sequence length and database size and is normalized based on the raw pairwise alignment score. So now we've just seen an overview of all the alignment algorithms available that's used for sequence alignment. Now coming back to our question, which of the following is not used for generating an optimal alignment of two nucleotide sequences? Option A, gap penalties. We saw that gap penalties has a certain score that is used to calculate the entire score of the alignment. And option B, match scores. We also saw that when two nucleotides match in two sequences, we have a specific score. So it's not option B either. And then option C, mismatch scores. We also learned that there's a certain weightage for sequence mismatches, and so it is used to consider an optimal alignment. So we rule out option C as well, and then that leaves us with the final option D, which is nucleotide composition, which is also the answer. So it doesn't matter what nucleotide the sequence is. For example, like let's say there's a match and the nucleotide at that position is A. It doesn't matter whether the match is between two A's or it's between two T's. There's no weightage for what nucleotide is at that specific position. So the composition of the nucleotide does not affect the alignment, the scoring algorithm. So hence the answer for this question is nucleotide composition.